can go ahead. I think we can definitely get started. I think that there will be people coming on in. Sonia from Dallas, Esther from New Jersey. People are still coming in for sure. All right. But I think that we are set, and I think that we can definitely get going here. So I do want to welcome you all to this awesome, fantastic talk on Nutrition 2.0, optimal health through the science of personalized nutrition. And, you know, what better person to give this talk? I'm always thrilled whenever Dr. Momo Vujicic, who is our founding chief science officer here at Viome and head of clinical research, when he has time to come on here with me and give a talk to our Viome audience. And specifically, I'm super excited on this topic here today, because this is what we have been buzzing about here pretty much the whole time that we have been here at Viome, but specifically recently. We just released a fantastic article on Nutrition 2.0. And I said, Momo, I want you to give a talk on this. I want you to give a live webinar talk on this. And he said, absolutely, I want to do it. And this is on Nutrition 2.0, meaning that nutrition for us as individuals, as we all have our own unique biology, and that's what's important for optimal health moving forward in the future. So I'm not going to give any more preamble. I've been talking too much. I'm just going to hand it over to Momo right now. Please take it away.
All right. Thank you, Lisa. Let's get started. So this has been my sort of focus professionally for the last 13 years. And prior to that, it was very much a personal focus due to some autoimmune issues I had developed early in my life. So this is really exciting times. So Nutrition 2.0 is a new paradigm shift in nutritional sciences that we've developed. We've been building for 13 years. And it comprises chemistry and mathematics. So everything that we do at Viome is chemistry and mathematics. There's no opinions, there's no subjective discussions or anything like that. Both are really objective, hardcore sciences that are based on data and objective equations. So we'll go over how we do that. So I have about 5 or 6 slides about some introductory concepts, because it's very important to understand these in order to understand Nutrition 2.0. So these are sort of foundational. So let's go over those. So first, the motivation. The motivation is that there are hundreds of millions of people today suffering from chronic diseases, and neither the academia nor the big pharma is actually out there looking for solutions. The academia are focused on publishing papers and getting grants, and pharma is focused on managing the diseases. They do not have any interest in curing the diseases, or they have very little interest in curing diseases. So I would categorize our current state of the art in the area of chronic diseases and cancers as dark ages, because back in the 13th century, a third of the world's population died of the plague. And today, we're not worried about the plague. We know what causes it. We know how it's transmitted. We know how to prevent it, how to cure it. And so we're not worried about it. We just read about it in history books. Yet 7 centuries ago, hundreds of millions of people died. Well, today, hundreds of millions of people are suffering from these chronic diseases, and they're as mysterious and bad luck as the plague was in the Dark Ages. And the very frustrating thing for me as a scientist is that we currently possess all science and technology to actually understand the root causes of all chronic diseases and cancers and prevent them, so that we can actually discuss them in the context of history books instead of suffering from them daily. And that's what Viome is about. That's why I changed my career back in 2010 and worked really hard for 6 years at Los Alamos National Laboratory to develop these foundational technologies. And now at Viome, we are trying to understand root causes of chronic diseases and cancers, and then enable people to prevent them, so that we're not getting those diseases and we're not afraid of those diseases. You guys probably all know, if you've read about nutritional advice, that nutritional advice is conflicting out there. If you ask a simple question, is coffee good for me? Well, you can find research articles that say it's good for you, and you can find research articles that say that's bad for you. Same thing for almost every food, you can find conflicting research. So what is it that you're supposed to eat if everything can either hurt you or be beneficial to you? And so we really need to put some science into this. One of the main wrong premises for the entire nutritional science so far is that it's based on population studies, which place all people into one bin and make recommendations for the entire population, but no one is the average. And so we really have to personalize nutrition. So let's talk a little bit about current nutritional advice. You guys have probably, I mean, many of you have probably gone and surfed the web and looked for all kinds of nutritional advice. I did this many years ago, and you find such conflicting advice, and you find these multiple different kinds of convictions. And so if you have a certain set of symptoms and you go out there and you seek advice from influencers, from bloggers, from nutritionists, from doctors, and you present the same set of symptoms, you're likely to receive a variety of nutritional advice that often is conflicting. Some people may recommend ketogenic diet, other people may recommend a Mediterranean diet that are essentially the opposite of each other. So that is not really a solid science if you provide the same input but receive multiple different outputs, because look at chemistry and mathematics. Chemistry and mathematics are very objective sciences. So what would you think of chemistry if you went to a lab to get your cholesterol measured and you went to 3 different labs and got 3 very different results? You wouldn't really trust that. Same thing with math. If you went to 3 mathematicians and asked them how much is 2 + 2 and you received 3 different answers, you wouldn't think highly of math. And so we are basically in that era of nutritional sciences where it's really not an objective science, which is why we built Nutrition 2.0. So let's just, this is one of the takeaway slides where I'm highlighting what are the Nutrition 1.0, the current state of the art in nutrition. What are the sort of, what is it? Nutrition 1.0 can address some common nutrient deficiencies, such as iron deficiency or vitamin D deficiency. However, if you have iron deficiency and someone gives you iron and you still have iron deficiency, they can't really help you. They don't understand the root cause of that iron deficiency, and they can't do anything except throw more iron at you, which may actually have significant side effects. Current Nutrition 1.0 often promotes fad diets, such as these ketogenic or Mediterranean or carnivore, or provides generic advice. Even nutritionists will provide generic advice such as eat more vegetables or eat more fiber. Well, which vegetables should I eat and which vegetables should I avoid? Well, nutritionists can't know that because they don't have the data for you. They don't know which vegetables are going to be good for you simply because they don't have any measurements of you. And then Nutrition 1.0 assumes that everyone is average and needs the same nutrition. And that's not the case. We already know that. That's now in the literature. That's been proven. We know that. So Nutrition 2.0 is what we've been developing for the last 13 years. Like I said, 6 years at Los Alamos National Laboratory and now 7 years at Viome. It is 100% data-driven and objective. It uses only chemistry and mathematics. From the moment we receive stool, blood, and saliva in our lab until our customers receive their recommendations, results and recommendations, there is no human intervention whatsoever. There is no opinion, there's no discussions, there's no human involvement. Humans were heavily involved in building the platform, monitoring the platform, and ensuring that the platform keeps improving by improving the chemistry and the math, mostly the math part. But there are no humans actually providing the results and recommendations. The results are measured by chemistry and recommendations are made using mathematical equations, 100% objective. Nutrition 2.0 is highly personalized. We have something like 450,000 customers and we have 450,000 diets. Our computers are not aware of ketogenic diet or Mediterranean diet. They don't know anything about it. They just know to fine-tune every person's diet to provide the best chance for their health. The goal of Nutrition 2.0 is to actually prevent chronic diseases and cancers and to slow down aging. It's not just, well, eat healthy. It's literally to fine-tune your body so you can be like that 110-year-old Japanese that is cognitively as good as a 60-year-old and physically as good as a 70-year-old. And then one of the most important parts about Nutrition 2.0 is that it's continuously self-learning and always improving. So whatever the state of the art today is in our platform, tomorrow it will be better, and the next day it will be better. And it's continuously improving until it reaches that perfection where for every condition and for every person on the planet, we know exactly what they need to eat, what lifestyle they need to lead in terms of sleeping, fasting, exercise, type of exercise, and so on. And we're getting there, and we're going to get there one day in a perfect state. All right, so now this picture is a short mental break because the next slide is going to be very, very technical. It's the most technical slide in the presentation. but it's really foundational to Viome and Nutrition 2.0 and our entire platform. So just enjoy this mountain view of the beautiful Pacific Northwest surrounded by clouds. All right, so here's the slide. So in many of our blog articles, in many of our talks, in many of our papers, you will hear the word pathways. And so I just wanted to really address this term, because if you don't have a PhD in chemistry, it may be daunting to understand what is this pathway. And you may hear only the shortcut pathways, but you may hear words like metabolic pathway or molecular pathway or regulatory pathway, enzymatic pathway, and there are probably other terms as well. I'm just going to shortcut it to pathway because there's some overlap. Some pathways are both regulatory and enzymatics, and all of them are molecular. Many of them are metabolic and so on. So let's just stick with a pathway. So here I'm showing sort of some of the most important pathways that we study. So think of this box here as a bacterial cell, and this bacterium is living in a colon of a human. And it just processed some fiber into glucose, and whether it processed that fiber or a neighbor processed that fiber, it receives a molecule of glucose. And because the colon has no oxygen, it cannot normally digest glucose the way it's normally digested. So it's going to have to ferment it. So it's going to ferment that glucose into butyrate. And in the process of using its own enzymes, every single one of these arrows is an enzymatic reaction that the bacteria perform inside their cells, and these enzymes are encoded by the bacterial genes. It converts glucose into butyrate, and during that process, it harvests energy in order to live. This is actually the food for the bacterium. And so when you see NAD or especially ATP, this is energy for the bacterium. So it converts glucose to butyrate, just like when you're brewing beer, yeast convert glucose into ethanol. This bacterium will produce butyrate, and then it'll release butyrate into the lumen of the intestines. And that butyrate has many functions that are very beneficial to us. And this is a box now where I'm showing a human cell. So this square box is a human cell, and it has receptors, which are proteins on the cell surface that absorb that butyrate, and that butyrate has following certain molecular pathways, usually regulatory pathways, will have actually an effect on gene expression of human cells. So it'll affect what our human cells do. So this is an example of chemical signaling. Metabolic pathway here produces a metabolite or a secondary metabolite or a chemical. You can also call it micropoop if you'd like, because butyrate is like a byproduct of bacterial metabolism. This molecule will now signal human cells to behave a certain way. So we are symbionts with our bacteria. We're not enemies. And one of the coolest effects that I personally have found for butyrate, even though it has many very cool effects, is that butyrate is a signal for T cells. T cells are one of the most important immune cells in our body that protect us from diseases such as the viruses, such as COVID virus, SARS-CoV-2. This is a signal for a T cell, once it recognizes, for example, SARS-CoV-2, to convert itself into a memory T cell, so that when we encounter SARS-CoV-2 or COVID a year later, our cells can get activated and say, hey, I've seen this before, I'm going to protect you from it. And so without this butyrate, our T cells may defend us against SARS-CoV-2 today, but they may not protect us a year later or 3 years later unless there's butyrate present. And so that's a chemical signal that our cells listen to and say, hey, I'm going to convert to a memory cell. So that's a very useful sort of an effect of a bacterial metabolite. All right, so done with pathways. Now it's all downhill from here. Let's talk about another concept, which is DNA versus RNA. You guys have probably all read genomics, or genetics. It's all DNA. I'm going to show you why DNA is static and boring, which is why the very first thing that we do at Viome, when every sample arrives at Viome, the first thing we do is destroy the DNA in that sample. That's the first thing, because it's static and boring. We don't want to work with static and boring things. We like to work with dynamic and relevant things, which is RNA. RNA is basically a copy of DNA that activates the gene. So the DNA encodes the genes, But once a gene gets activated, it gets copied into RNA, which activates that gene into protein and function. And that's why we study functions. We don't study the potential. So we're going to give a whole new webinar on RNA versus DNA and how Viome is different and talk about that, but just give you sort of a taste of that. Let's talk about, you order a DNA test, your own genetic test, and the test results come back for Crohn's disease and say, uh-oh, you have an increased risk for Crohn's disease. And you're like, wow, that's not good, that's bad. Well, not necessarily, because the vast majority of people who have this increased risk for Crohn's disease will not, in fact, develop Crohn's disease ever in their entire life. So sure, it's an increased risk relative to other population, but it doesn't mean at all that you're going to develop Crohn's disease. In fact, you likely will not. There's nothing really you can do. This doesn't tell you absolutely anything about what can you do to prevent Crohn's disease. So it's not really helpful in prevention. And if you do develop Crohn's disease, this test still doesn't tell you what to do about it. And so this test is basically like a game. It tells you you have an increased risk, and that's it. Scare yourself, but there's nothing really actionable here. So that was sort of the state of the art back when I started this journey of developing, epigenetic assays with RNA, that I realized this is nearly useless. I don't know what to do with this information. It's not going to help me. We really need to shift from genetics to epigenetics, which is where RNA plays the big role. And this is another takeaway. I really would like this sort of to be imprinted in most people's, retinas, is chronic diseases and cancers are not bad luck, and they're not a genetic fate. There are very few diseases called rare genetic diseases where if you have a genetic mutation, you will develop the disease. There are very few of those. The vast majority of chronic diseases and cancers are not genetic fate, and they're certainly not bad luck. They're simply a result of chemistry that we do not yet fully understand. That's it. It's just like the plague was in the 13th century. They didn't understand what it was. It killed so many people, but we now understand it, and we know that it's not either bad luck nor genetics. It's just we didn't understand the chemistry, and now we do. So this is the key. Everyone should be seeking knowledge about what is the chemistry that can lead to chronic diseases and cancers, and how do I reverse that chemistry with healthy eating and healthy lifestyle. 13 years ago, when I started talking about it, everyone looked at me like I was crazy. But now there's actually very solid scientific literature showing that genetics plays a minor role in chronic diseases and cancers. There are now many studies on genetically identical twins that show that genetically identical twins do not develop same chronic diseases and same cancers. In fact, there's a very minor role that genetics plays in the chronic diseases and cancers. And this has been very well quantified on a very large study epidemiologically. So you can go out and read that, and you can go and look at these two references, but there are many. So let's dispel these myths. I'm just going to give you a teaser here about our metatranscriptomic method. Metatranscriptomic is just a technical term for RNA sequencing. We analyze RNA, as you guys probably know. And this is a comparison slide, how big of a quantum leap metatranscriptomics is over the other methods of analyzing, for example, the microbiome. In addition, metatranscriptomics gives us access to human gene expression, not just microbial. And so there are huge advantages, and we will discuss these in detail in another webinar, but just wanted to show this slide to have it there. Here's another takeaway. This is a very important slide, and it goes back to that butyrate and so on. And so our body does not understand microbe names. So when you do a genetic test of your microbiome, for example, and they tell you you have Faecalibacterium prausnitzii, all that test tells you is you have this bacterium. And everyone knows that Faecalibacterium prausnitzii can produce butyrate. It can, but it doesn't. It doesn't always produce butyrate. So it can, but you don't know if it does. And so let's go over these bullets. Our body does not care that we named a microbe Faecalibacterium prausnitzii. If my T cells are encountering a virus and they want to convert to a memory T cell so that I have lasting immunity, my T cells do not understand. They do not have a brain. They don't understand the concept that we have named a microbe Faecalibacterium prausnitzii. That doesn't have any impact on my immune system. Our immune system, my T cells, also do not understand the fact that Faecalibacterium prausnitzii contains genes for producing butyrate, and it's capable of producing butyrate. That doesn't make any difference whatsoever. The only thing that matters to our body is is butyrate actually produced? And a genetic test, meaning a DNA-based test, will never, ever in history of the world be able to tell you whether that bacterium is or it isn't producing butyrate. It's only telling you there's a potential for it, but I don't know if it is or not. And this is why we don't measure DNA, because you just don't know whether it's doing something or not. Our RNA test tells us whether Faecalibacterium prausnitzii and other bacteria are actually producing butyrate and how much butyrate they're producing using those pathways that I explained by actually quantifying the activation of the genes for production of butyrate. We do not model that. We do not assume. We do not infer. We do not talk about it. We measure it with a quantitative, clinically validated tool. And that's the only way to do it. If you're going to do something, do it right and measure it. Don't talk about it and predict it and assume things. So this is a really key takeaway, one of the key takeaway slides. All right, so how do we do all this? How do we do chemistry and math? We do it using our systems biology platform. So systems biology just means that when we're talking about any human condition, whether it's brain related or muscle related or gastrointestinal tract related, we assume that many systems in the body are involved in that process. And for example, to give you a sort of a very egregious example, bipolar disorder for decades has been considered to be a brain disorder, and all the drugs target the brain chemistry. We now know that that's very unlikely to be the case, and that in fact, the gut microbiome is the main key role player in the onset and progression of bipolar disorder, because if a person with bipolar disorder performs fecal microbiota transplant, they can completely cure themselves without changing their brain, without changing their genetics, without changing their diet or anything else. They simply just swapped out their microbes with someone else's microbes, and boom, bipolar disorder goes away. So systems biology is really looking holistically at the whole body, and we want to digitize the whole body, and we are using this platform called ViOS that has that chemistry and math. So this is Viome Operating System. So think of it as iOS, but ViOS. It's a very large technology platform that, you know, we give 8-hour presentations on that technology platform, but here's one slide about it. So what is ViOS? ViOS takes data from as large of a population as we can gather in the whole world. And currently in our database, we have data from 450,000 customers and 16,000 research participants. And we collect molecular data. These is the chemistry from stool, saliva, and blood, and some other sample types for research purposes as well. And then we collect comprehensive metadata. And metadata are, you know, anything about their use of medications, supplements, their lifestyle, their symptoms, their diagnosed diseases, their anything we can learn. And then we use machine learning to generate these mathematical models of different conditions, diseases, and so on. And phenotype means a condition or a disease, or even like physical fitness or cognitive fitness or anything that we want. We use machine learning to produce these mathematical equations. So this graph represents essentially a mathematical equation where each bar here is what we call a feature, which is a data point. Is it a microorganism species, or is it a microbial gene, or is it a human gene, and how much it contributes to the condition we're studying. And this is positive association with that condition, and this is a negative association with that condition. So this is actually a mathematical formula plotted as a graph to be more easily understood. And so we develop these mathematical equations for every condition, every symptom, every disease in the world from a very large data set, and we continuously learn. We do machine learning 24 hours a day. And systems biology obviously must include microbial components of the human body, and this slide just shows you that we, as a human physical body, we only have 23 genes that are encoded with our own genome that come from our parents, but the microbes on our bodies encode about greater than 2 million genes. So you can see that roughly 1% of all genes encoded in our body is actually human. The rest are all microbial. So we have to study the microbe very heavily in order to understand our physiology. All right, so that was all sort of a introduction and foundational concepts. So now we're going to get to the meat of the presentation, which is Nutrition 2.0. So Nutrition 2.0, as a recap, is we take a person, we digitally profile them using molecular data from multiple samples, stool, blood, saliva. In research purposes, we also do urine and plasma and throat and respiratory swabs and all kinds of other samples. We measure chemistry using clinically validated molecular assays. And I cannot overemphasize clinically validated. Most of the tests out there for microbiome are not clinically validated. They're research grade tests, which means that you don't know if you provide the same sample 3 times if you're going to get the same result, because if a different technician processes it or different reagents are used, you may get 3 different results. So really, you should demand that whatever test is performed on you, that it is clinically validated and it is performed in a clinical laboratory that is licensed by the United States HHS, Health and Human Services. That's like a minimum requirement. And then we overlay mathematical equations that we learned from all of the data we have to produce personalized nutritional recommendations for health. And we already went over these bullets, so I'm not going to do that again. In terms of RNA analysis, I just want to, we're going to go over this in more detail in our next webinar, but what does RNA analysis provide? It provides composition, also technically known as taxonomy, and functions. All of this is what I call chemistry. So we obtain genome or strain level composition of the microbiome of all microbes. That includes bacteria, archaea, fungi, which includes yeasts and molds and so on, and any kind of a virus, whether that's a human virus, whether that's a plant virus or microbiome virus or bacterial or fungal virus, we see them all. We see microbial gene expression. Those are those pathways that we talked about. We see human gene expression, and we see, very importantly, human microbiome interactions. And these human microbiome interactions, we plot in these massive, massive heat maps that our machines generate and that we interpret. And this is where human expertise of biochemists and microbial physiologists and nutritionists and computational bioscientists and computational systems biologists all come together to interpret this and put together these mathematical equations that we use. Where do our mathematical equations also come from? They come from these large studies. I already told you guys that we have enrolled so far 16,000 clinical research participants in our programs, and that's where a lot of our equations come from. And I'll just highlight one of our programs. We performed 2 clinical trials in the United States and Japan. So we split it halfway. So we enrolled 550 people in the US and 550 people in Japan. We fed those 1,100 people a total of 60,000 meals. We provided 40,000 of those. We had truckloads of food coming from Whole Foods, and we were providing those foods to our participants. 20,000 they consumed on their own, but they told us exactly what they ate via an app. And so for 14 days after we collected their samples, for 14 days, they ate these 104 very carefully prescribed meals, designed meals that have a lot of different variety of macronutrient ratios in terms of carbs, fats, and protein, very nicely designed by our nutritionist team. We measured their glucose response to every single meal over that 14-day period using continuous glucose monitor. So now what we put into this supercomputer is 60,000 meals to 1,100 people and measure and provided the glucose response to every one of them. And we asked the computers, can you actually have used the molecular data that we collected prior to this study to predict the glucose response instead of having someone measure themselves for months and responding to different meals? And the machines came back and said, yes, we can build a very accurate model of this. And so we built that model, we published it, we incorporate it into our app, and every single customer in the world benefits from this. This is now integrated in our app. This is what we call the math. We measure their chemistry using our tests, and we overlay them with mathematical equations to know exactly what you should be eating and what you should be avoiding so that you can minimize the glucose response in your blood. This is just one of many, many studies that we have done at Viome. I'll just highlight a couple more. We did a very large study where we wanted to understand what are the molecular determinants of the onset of prediabetes and diabetes. We were able to build state-of-the-art models, and I will show you some efficacy later. We have also built these scores. So every customer sees scores in their app. These scores are all based on those pathways. Almost all scores. There are some scores that are based on just the presence and absence of some microorganisms, but the vast majority of our scores are all based on pathways. And you can see here, we have an oral inflammatory pathway score that we've built. And you can see in blue here, these are people who have self-reported to have some kind of an oral or health issue, periodontitis or gum disease or gingivitis and so on. And these are people who say that they do not have that. And you can see how much higher the score is for the people who have something, and the p-value for that difference is massively low. So this is a very statistically significant difference. The same thing here is for general metabolic health. And so people who have worse oral inflammatory pathway score, they generally have worse metabolic health. And this is now not an unknown thing. This is actually now known in the literature. There are other publications showing that gum health is very important for systemic health, and we have that built into our scores. Let me go to the next slide. We have methanogenesis pathways, and here we're showing, let me see if I skipped one slide. Yes, we have sulfide production pathways. Remember, pathways are those molecular pathways that are producing hydrogen sulfide gas. When these are high, people have increased motility or diarrhea. And you can see here that people who score high for increased motility or diarrhea, they have a high sulfide production pathways. People who say that they have low motility, they have low scores, and people who have healthy motility, they have medium range scores, and the p-values are here. These are super statistically significant differences. And we measure these pathways, we quantitatively measure them, and we actually provide, our computers provide each participant, each customer with specific dietary recommendations to optimize this score, as well as all other scores. We have methanogenesis score, which is the opposite effect of sulfide production score. Methanogenesis is associated with constipation or low motility. And you can see here with super high or super low p-values, we can distinguish people who have normal motility, high motility, and low motility using methanogenesis pathways. And this is many pathways in many different microorganisms that contribute to methanogenesis. Okay, so the next question is, okay, this is a great platform. Does it actually work in real life? So we've been working now on this over the last year and a half or two, and I'll just give you a highlight of what we've been working on. First stage was we asked the question, okay, does this work in our customers? And so here we took 1,500 customers who self-reported as having low adherence to Viome, and then 1,500 customers who said that they had high adherence to Viome, and we basically measured their type 2 diabetes score that we developed. And we show here that even low adherence Viome customers reduced their type 2 diabetes score, risk score, but the high adherence customers reduced it much more with a very low p-value. So customers who followed our recommendations more, they benefited far more in terms of reducing that risk score. Here we go with depression. So here what we did is we divided our customers who had depression, different levels of depression, into those that not only did not follow our PS, which is personalized supplements, but they also stated that they did not follow our diet recommendations. And we compared them to customers who consumed our personalized supplements and they said that they followed our dietary recommendations. And we asked the question, well, was there an effect on symptoms of depression, which we call mental health here, but it's quantified with a clinically validated survey for depression called PHQ-9. You guys can look up, all this is used in clinical practice. And what we are plotting here is, this is the initial profile of Viome customers who consumed personalized supplements. You can see that some had severe depression, some had moderate depression, and some had mild depression. And after a period of a few months, you can see that the numbers of severely depressed decreased, moderately depressed decreased, and low depression decreased. And the biggest number now is people who have no longer symptoms of depression at all. And then if we look at the customers who had low adherence, you can see that severe depression increased, moderate depression decreased, low depression stayed the same, and some people went into low depression. But the statistical difference here is huge. The p-value is less than 0.001, and the effect size is 0.49, which is a very good effect size. So here we're showing some preliminary pilot data from our customer population that our personalized diet may actually work very well. So that's a very nice finding. Now, the gold standard for knowing whether an intervention works or not is to run a randomized placebo-controlled trial. And so we are currently actively running 4 randomized controlled trials. So this, I'm the director of clinical research. And so we are running these 4 trials for depression, IBS, obesity, and type 2 diabetes. I believe that depression has already completed the enrollment. So we're close to being done. Hopefully in a few months we'll be done. These 3 are actively recruiting. If you guys are interested, you can spread the word about these trials. And so we will test here, how well does Viome Nutrition compare to placebo pills that have obviously should not have any effect. So we're very excited to see the results of this. The audience is welcome to also read our articles. We have a lot of articles that we have published. Here's a list of peer-reviewed articles that are in the literature. Here's a list of some very exciting articles that are now under peer review, but they are all available as preprints. I also encourage you to find our blog. We have published now a few very, very nice blog articles with actual data from our database, actual score descriptions. We have a beautiful scores blog article. We have another blog article about Nutrition 2.0 that summarizes sort of this webinar. So please inform yourself. And that's it. That's the technical presentation. We are in 2 locations in the Seattle area, and we have a location in downtown Manhattan. That's where our bioinformatics, data science, and computational systems biology teams are. Welcome to, I welcome any questions now.
Yeah, 2 beautiful cities here that we operate in. They're awesome. Wow, Momo. Okay, so I do want to start with something. Irina in our audience was asking, referring to the study material that you were showing us. What does p-value mean? If you want to flip back up in the
yeah. So p-value is basically the percent chance that something happened by random chance. And so if you measure many different things about a person and try to predict whether that person has some phenotype or not, then if you measure a lot of things and have very few people, by random chance, you can pick out something that you measured that is highly correlated with the phenotype, but it's actually not true. And so mathematically, you can compute that, and we compute that. So the generally accepted p-value in the scientific literature is 0.05, meaning that any p-value below 0.05 is statistically significant, and 0.05 you can translate into a 5% chance. So if you divide it by 100, that's 5% chance that there's a 5% chance that this happens randomly. And so the lower the p-value, the lower the chance. So when we displayed one of the p-values that was 1 × 10⁻49th, I mean, that's an extraordinarily low chance that happened by random chance, that's likely a very truthful result.
Okay. All right, good. So Irina, I hope that helps you. P-value. All right. So there are a few instances of people asking about how do we find out more information about signing up for a study. So Momo, with your permission, I will put those people directly in touch with your team. I will directly get them in touch with your team so that they can find out more information about that. Because
Lisa, they can also, we do have an active and dynamic website. If they go to viomelifesciences.com, there is a tab for research, clinical research, and they just need to go there and look at all the active studies, because we have more active studies than just the 4 trials. And that's a dynamic page, and it'll be updated as more and more studies. So that's really something that if people are interested in participating in research, please go there.
Yeah, viomelifesciences.com. Don't want to stutter on that. Excellent. If you want more information for anybody, or if you know somebody who might be interested in joining one of our studies. All right, I do have a few questions here, so let's do it. John is asking, you did talk about what research is being explored right now at Viome, obviously with our studies, but also he asks, what are the limits of AI in the Viome universe?
Currently, the limit is data and money. There are, with Amazon Web Services, computational power is basically limitless. I mean, if I don't know how much money we would have to drop on Amazon for them to say, okay, we are out of servers for you. Right. So that's really nice, because that was the limit for many, many years. And so it's really the data. We need as much data. If you look at a complex disease such as depression or IBS or type 2 diabetes that has many, many root causes, it's not like an infectious disease where you have one bug associated with one disease. That's a very simple problem to solve. We do need a lot of AI, but there is currently, the limitations are, like I said, our data, the amount of data we generate, and the limitation is how much money we can give to Amazon for all the computational analyses.
Yep, have to pay the servers, have to pay the servers. All right, thanks, John. Mateo asks, can we say that with the epigenetic testing with estimated risk results, we might be doing subclinical diagnosis? I thought that was kind of an interesting question.
Yeah, that's an interesting question. I don't think so. I think that that genetic test has nothing to do with the actual systems biology state of your body. In other words, someone with a high risk score for some disease could be 100% molecularly healthy, meaning that none of the pathways that eventually lead to that disease are actually activated, and all of the protective pathways that protect us and prevent us from developing that disease are actually activated. And so I think that I really don't, I would not associate with that high risk score with any subclinical scores or molecular pathways. It's just, I wouldn't. I think that that's simply a potential for that risk at birth, and it doesn't change during the course of your lifetime, regardless of how your molecular pathways change. It's a good question. We could have a debate on that one day.
No, absolutely. Okay, so it looks like this next question, and I think we can field this question. This is kind of more a guru's area, but what is the biological age result based on? Since we were just talking about AI.
Oh yeah, I know what
you know.
what guru knows. Okay, so what we fed AI was, the first model was, we fed it all of the chemistry data from stool only, which means that I listed that on one slide, which is all of the compositional components of the human stool, meaning the relative abundance of every single microbe in the stool sample. Next, we fed it the gene expression level of the microbial genes. Then we fed it the gene expression level of the human genes. Even though it's stool, there are in fact human genes that are expressed in stool because we shed epithelial layers into stool, and the expression level of those genes can be different with age. We fed all those data, and we provided the computer with the actual chronological age and asked the computer, okay, which of these features that we fed you from all the molecular data are the best at predicting the chronological age? And it turns out that all of them are predictive. There are some that are microbial composition. There are some that are human gene expression, but the vast majority are the microbial functions. We did find one human gene expression that was so highly correlated with gene that with age that we think it's a potential therapeutic target, but when we're going to get into therapeutics, that's a good question. I don't know. So yeah, all of those. And now we've developed the next generation of aging model, which is based on stool, blood, and saliva. So we provided the model with all molecular data from all three data sets, meaning all of these features in stool, all of the features in saliva, and all of the features in blood. And please note that because we're quantifying all RNAs in blood, that doesn't only include human RNAs, that also includes the minor component of microbial RNAs that come through either leaky gums or leaky gut or some other place into the bloodstream, and we actually quantify those as well. So we gave the machines all of those data, and we asked, okay, build us a model, and the machines built a much better model than just based on stool. But it is, again, a set of compositional features in the microbiome space, and then gene activity features in the human space, and gene activity features in the microbial space. And we are preparing a publication for that right now, so that'll come out whenever peer review ends, which is uncertain.
All right, thank you. All right, so this next question. All right, so if Nick says, so if your BIOS is changing based on new data, is it possible that my next month's supplements may have changed based on data other than my test results?
Okay, so yes, I wouldn't say next month. We don't change the mathematical equations every month anymore.
Right.
But well, speaking of next month, actually, next month we are going to have a major ViOS update. Major, it's one of the major updates. But I would say going forward, we're going to have maybe a major update to ViOS equations once a year or so, and then fine tuning in the meantime. And that fine tuning is not going to be necessarily dramatically changing anyone's recommendations or supplements. It's going to be fine tuning. The major changes would be if we, for example, run a very large study on a particular condition, let's say IBD, and we come up with new mathematical equations to help people specifically with IBD, then only those people, when we make the upgrade, would actually potentially see larger changes in them. So it depends. It's a complex thing, but I think that summarizes it. That going forward with minor changes, no one will really notice big differences. With major changes, a subpopulation of our customers that have specific phenotypes where mathematical equations have changed, they will potentially see a significant change in their results and recommendations.
All right. Excellent. Okay, so next question. Lisa asks, what actions is Viome taking to encourage acceptance of your modalities in mainstream medicine, if any?
Yeah, I mean, we are publishing papers. We are going to conferences. We do speak with doctors a lot. Our chief medical officer is a director of an endocrinology clinic at Advent Health. And at Advent Health, they now have a lifestyle medicine program. And the first thing that they do at his department when someone comes with pre-diabetes is they ask them to diet change. They don't give them metformin, they don't give them any medications, they try the diet approach, and they have classes with chefs at the clinic. So that's one thing. Second thing is one of our scientific advisory board members is actually organized a lifestyle medicine program at Johns Hopkins, and we're trying to do as much as we can, but there is no nutritional training for doctors in medical school. And so when they come out of medical school, unless they pursue lifestyle medicine as their choice, they're not really going to be well equipped to be able to promote the nutritional ways. So we're doing everything we can, but we're a small company with 140 employees, and we're doing as much as we can everywhere we can, but it's going to happen one day.
Absolutely. Small but mighty, we are. Absolutely. Jennifer asks, do you plan to do any studies in autoimmune patients like MS, lupus, RA, Hashimoto's?
As soon as we possibly can. I mean, all of those are super interesting, super important. All of those listed. Yes, as soon as we can. It's just, we have to be very careful as a startup that's not profitable. We have to be very careful where we invest money into research dollars, and we have invested those in the most prevalent diseases. And as horrible multiple sclerosis is, it's simply not prevalent enough for us to make that investment now. So we'll have to wait until we're profitable, and then we'll make investment. But one by one, we will cover every single chronic disease and every single cancer. As long as I'm alive, that will happen. There is no doubt about it. I wake up every morning, 7 days a week, and work on it.
Absolutely. Absolutely. All right. So I have this question here, and I think, and I just want to make sure that we address it. Chelsea says, I'm not seeing any of these things in the app right now. Is this something that's coming, or do I have to update the app? She's referring to Nutrition 2.0, but I just want to let you know that what we are presenting to you today is what we do. It's what Viome does. We test your samples, we gauge your health right now according to your samples, and we match you up with the best foods possible for your biology right now using Nutrition 2.0. So what you see in your app is what we do with Nutrition 2.0. So there's no updating that's necessary. Just keep on using your recommendations and retest when necessary. Is there anything else that you would like to add, Momo?
No, I think that's a perfect answer. That's exactly right. None of what I said is speculative or future. This is what we have been doing and what we do today, everything. And so, yes, thank you for asking that question. This is what we do.
Awesome. And all of you who are asking if there's a recording to this, absolutely. Everybody's going to be getting a follow-up email tomorrow with the link to the recording. So you can not only watch it yourself, but you can pass it on to somebody else who needs this information. All right, so let's see. Chris is asking, do you have to physically reside in one of your locations to participate in one of the studies?
No, absolutely not. All of our studies most of our studies currently are direct to participant, and we have shifted to that model because working with hospitals and CROs is either too expensive or too complicated or both. So most of our studies and all 4 of our randomized control trials are direct to participant. The only limitation, unfortunately, is that we're limited to studies mostly in the US. The only way we run studies outside is with partners. So we just completed a study in Australia and New Zealand, and we have 2 studies in Australia, and we have a study in Brazil right now, and we ran that study in Japan. But those are all with partners. But the vast majority of our studies are in the US alone. If someone can take on the ability for the United Nations or the World Health Organization to provide an ethical committee service for researchers like us, where if you get an ethical approval by them, you can perform studies in all countries in the world, that would accelerate this kind of approach massively. Because if I have to go and get ethical approvals in 40 other countries, we would literally never do any research because I would just be bogged down by bureaucracy. That's all my team would be doing. And so we can't afford to do that. And so if someone can figure out how to do that at the level of UN or WHO, that would be a huge contribution to science to get rid of 99% of the bureaucracy so we can focus on science.
Yep. So if anybody has connections and we're serious about this, please let us know.
Passion is all it takes. You don't need connections. You just need passion.
Yes, passion. Excellent. Last question I'm going to give to you, Momo, before we end off. And I do have a special gift for everybody who's here today. Robert asked, do you have any insights on the relative importance of taking the Viome supplements versus following the diet only? So Viome obviously recommends foods as well as specific supplements, and he's asking foods versus foods and supplements.
Okay. So first of all, think of foods as supplements. Think of them as chemicals, or sorry, I said that wrongly. Think of foods as chemicals and think of supplements as chemicals. We need a certain flood of certain chemicals in certain amounts in order to feed our cells properly, to maintain our pathways healthy, and to feed our microbiome pathways healthy so that they can make us healthy. And our computer algorithm is such that it prioritizes the provision of those chemicals from foods. Only when there's a conflict in foods does our computer then recommend a supplement. So for example, if a chemical is present in certain foods like broccoli that's beneficial for you, but broccoli also has a detrimental chemical for you, then the computer will say, well, wait a minute, can I provide that beneficial chemical in a form of supplement? If I can, then I'll say avoid broccoli and just take the supplement. Those are the kinds of conflicts. Or when someone needs to take enough of a chemical such that they would need to eat a pound of blueberries every day. It's unreasonable to ask someone to eat a pound of strawberries or blueberries every day. So then, oh, can we replace that with supplement? Yes, we can. And so I absolutely have no way of saying that foods are more important than supplements. They're all extremely important, both of them. So I would say follow both to the best of your ability. Obviously, supplements are much more easy to follow because, well, you can just buy them and eat them. Foods are much more challenging to follow, but just do your best.
Exactly. Do your best. All right. Thank you so much for answering that last question. And we are pretty much at time. I want to thank you, everybody out there in the audience, for joining us and spending the last hour here with us. And I want to thank Momo here for giving us a really great talk on Nutrition 2.0 and explaining to us how this works with Viome and how you can put it to work for yourself and optimizing your nutrition and giving yourself better health. And I also want to extend to everybody a special discount code for July webinar, one word, July webinar, and you can get $100 off our full body intelligence test. If you haven't tried it yet, if you're up for a retest, you want to check in again, go to Viome.com and use that discount code. It's good through July 23rd, I believe, Sunday, and order yourself a full body intelligence test. And get on board with Nutrition 2.0. Thank you again, Momo. Appreciate you spending the time with us and giving us this talk. And we will see everybody again here very soon for our next live webinar event. Bye-bye.
Bye. Thank you, Lisa.
Thank you.